Good afternoon, my microbiology students, and welcome back. This is part 2 of chapter 8. So previously, in our first part of this video, we talked about genes and genetics. We talked about some key terms. We talked about the structure of DNA. We talked about the structure of DNA in comparison to eukaryotes and prokaryotes like bacteria. And we talked about transcription and translation, which is how organisms make proteins. So next, what we're going to talk about is DNA recombination events, where DNA can be transferred from one bacteria to another. And so, therefore, because DNA is transcribed into mRNA, which is then translated into proteins, that means if we give new DNA to an organism, they're now able to make novel proteins. And this is really important for bacteria to be able to do because they don't have an exact equivalent to sexual reproduction. So if you recall several chapters back, we talked about how bacteria reproduce by a process called binary fission. And as a result of binary fission, the first cell in a generation will split to form two cells that are genetically identical, unless there's some sort of mutation. So that means that just via binary fission, there is not a whole lot of genetic variation in a population of bacteria, and that's not really good. Think about it compared to humans. We do sexual reproduction, and that's one of the many ways that we produce genetic variation in our population. So in order to increase genetic variation, these bacteria cells do have their own ways of sharing genes, and this sharing of genes is called recombination. They're recombining their genetics. So I like this little meme here. It shows here we have a little blue guy that's a bacteria, and he's like, ooh, I made this. It's one of his little chromosomes or plasmids, and he's saying, here, you can have it. And so now that he has it, he turned blue, because remember, DNA, which converts to RNA, which eventually then will convert to proteins, is what gives us our traits. And that was part of that conversation of genotype and phenotype. Genotype being the genes you have, and the phenotype being how those traits physically manifest. Just like we all have different hair colors, and it's because we have different kinds and different amounts of melanin, which is a protein that we produce, and the amount and kinds of melanin we produce is dependent initially upon what our DNA says, our genotype. So, again, sharing of these genes between bacteria cells is called recombination because we're recombining DNA from two or more different organisms. So, why is it useful for bacteria? Sometimes these genes that they trade amongst other bacteria can provide resistance to drugs or some kind of other metabolic poison. For example, penicillin is resistant to Staph aureus is resistant to multiple different types of antibiotics like methicillins and penicillins and others. So originally, that wasn't the case, but what we're seeing more and more of in the current day and age is that more microbes are being more resistant and more resistant to the drugs that we currently have. So they can also provide genes for new nutritional and metabolic capabilities. For example, some microorganisms are capable of utilizing lactose, which is sugar, to drive its metabolism, to make energy. But not all can. But if there is a lactose metabolizing gene that was given to a different organism that lacked it, then they would now be able to consume, or not consume necessarily, but metabolize lactose. They can also increase their virulence, how effective they are. And also these new genes can help them to be more adaptive to the environment that they live in. And another reminder, one more time, what is recombinant? It is any organism that contains genes that originated in another organism. So this guy right here would be example of a recombinant organism. Because, of course, he has his natural chromosomes or genes that he has, but he also was donated this chromosome here, or plasmid, from our original guy up here. And that's why he turned blue. So it would be my guess that this cartoon is showing that this plasmid he's donating to this green guy is a DNA for the color blue. And so now he's like, ooh, I'm blue. So it is a recombinant organism because he has DNA that derived from a different organism. Alright, so how do these bacteria actually do this transfer? So all of the types of transfer we're about to discuss are called horizontal gene transfers. And in comparison, if I were to describe vertical gene transfer, that would be genes being passed from parent to offspring. But in the case of bacteria, that's not what we're seeing here. We're not talking about genes being passed from parent to offspring, because that would be binary fission. And as we've said, that leads to genetically identical cells. They have to have other means of increasing genetic variation. So instead, we have horizontal gene transfer. So another way you might be able to think of that is, here would be an example of vertical gene transfer, where we have parent and offspring and offspring. I don't think I'm able to fit all that in there, but you see what I'm saying? I should have shortened it to just the O, but this would be vertical transfer. Now, let's say I have a different initial cell over here that's different parent and its offspring. Alright, so now if this particular organism has a gene for, let's say, penicillin resistance, so I'm going to write penicillin resistance, its offspring will also have that gene. My P looks funny there. Sorry. Also has the penicillin resistant gene. So horizontal gene transfer means that it's transfer in the same generation. This would be generation 1, generation 2. These organisms are not related. Okay, so like this is a relationship here, parent offspring, and this is a relationship here, parent offspring. So in horizontal gene transfer, we see transfers of genes between organisms in the same generation. So this bacterium could pass its penicillin resistant gene to a member of the same generation. And how they do this is by exchanging plasmids. And so as a reminder, here's my little bacterial cell. And we have my single circular loop of DNA. And loop there, perfect. And remember, we have plasmids. Alright, so plasmids are chromosomes that contain accessory genes, meaning they're not vital to life, but they do give us some interesting traits. So they're small accessory rings of DNA. They're not necessary for survival, but they can carry useful traits. So how do we trade these plasmids from one microbe to the next? There's a few different methods, and they are called as followed: conjugation, transformation, and transduction. We'll start with conjugation. Conjugation is the exchange of DNA from a donor to a recipient cell via direct contact. Direct contact. And this direct contact is due to a conjugation or sex pili. So in gram-negative cells, this is specific to gram negatives, there the donor cell will have a fertility factor, and that fertility factor is a gene. And it allows for the conjugation, or the synthesis of what we call a conjugation pili, or pilus is a singular, pili is plural, so just to clarify if you hear me say something slightly different. And then the recipient cell will have to have a recognition site for that pilus. And so as a reminder, what a pili or pilus is we have, this was in I think the eukaryotic structures chapter, but it's where they can form these little bridges, these like tubes between two bacterium. So that serves as like a little highway here. Here's my chromosome for bacteria 1. Here's my chromosome for bacteria number 2. And let's say this is the plasmid that was going to be exchanged. This little sex pili, or conjugation pili, I'll call it a pilus here, singular, will now be able to freely be transported from cell 1 to cell 2. So this pilus is kind of like a little highway for gene transport, where this would be the donor and this would be the recipient. So the pilus grows out of the F factor positive cell, which was the donor. This one has the fertility factor that allows it to form that pilus. And it attaches to the surface of the F negative cell. So I'll write in F positive, F negative. And it is going to contract and draw those two cells together. Now, it does get a little bit more complicated because that can actually happen in more than one way, as we're about to see next. So the donor cell can sometimes make a copy of its F factor first and transmit this to the recipient, which was previously F negative. Well, that would then turn that F negative cell into an F positive cell, which means that second cell is now capable of producing its own pili, or own pilus, and do conjugation with other cells. The second way that this can happen is called HFR donations, or high frequency recombination, and this is where a plasmid will become integrated into the fertility factor positive donor cell's main chromosome. Then, when replicated, it can begin transfer to a recipient cell via the same mechanism, the pili, but in this case, some of the chromosomal genes are transferred to the recipient and not just the plasmid. The plasmid genes may or may not be transferred. So it's kind of interesting how differently it can take place, but overall it's the same process, just slightly different subtle details. So if we look at the picture here, I think it'll probably all come together. This is showing you the regular F factor transfer. So we see a donor cell and the recipient cell. Again, the donor cell is F factor positive. That means that it is capable of making this pilus. This cell is the recipient cell, and it's F factor negative. So it receives the pilus, but this cell on its own cannot make a pilus because it does not have F factor. So now, if F factor is found on the plasmid, and again, this would be the main chromosome. This multicolored one is the plasmid. Main chromosome. Here we see that the F factor, the entire plasmid that it's on, has been replicated, and one of the copies is going into the recipient cell. So now, after all is said and done, it's essentially like this cell number 1, the donor, has given the recipient the ability now to make a pilus and connect with other cells for future gene transfer. On the other hand, the high frequency transfer, this is where we are going to have a donor cell and a recipient cell still. We see the plasmid, though, is integrated into the chromosome. So in comparison, here the plasmid was separate from the chromosome, but now we see the donor's plasmid integrated into its chromosome. So it can still form a pilus because it has that F factor here in the plasmid. So whenever the chromosome is replicated, maybe there's a partial copy of the donor's chromosome and some of the plasmid, which is what this is showing. Is it going to copy just the plasmid and send it over like it did in this scenario? No, not necessarily, because it's integrated into the main chromosome. So it's very likely that not only are we going to copy and transfer a copy of the plasmid, but also part of cell number 1, the donor's natural chromosomal genes. And so now we have our first cell as normal, the donor's, this is the same as this. But if we look at the recipient cell, it has its natural purple chromosome, but now also donated genes from our donor, DR, donor and recipient. And in this case, it does have the plasmid DNA, but it also has some of the chromosomal genes from our donor cell. So in comparison, in this scenario, we saw just the F factor or the plasmid being transferred from the donor to the recipient. But here, because the plasmid was integrated into the main chromosome, some of that main chromosome was transferred to our recipient in addition to the plasmid. So that is HFR transfer. So now not only is it able to make a pilus because the F factor got transferred, but who knows what these other genes do. It's getting given traits now that this first bacterium had that this one previously didn't. Now that's for gram-negative cells. What about gram-positives? So in gram-positive conjugation, there is an opening that will form between two adjacent cells, and replicated DNA can pass from one cell to the other. So this process is conservative in gram-positive organisms because the donor bacterium will retain or conserve a copy of the genetic material that's being transferred, so it doesn't lose anything. So again, why is this important? Because it gives these bacterium some interesting traits. And we like to talk about this in micro because it's a huge problem, but this can account for a lot of the mechanisms behind which microbes are becoming more and more resistant for various antibiotics or any other antimicrobial substance. And this trait for being resistant to various things oftentimes is a process through sharing resistant genes through this process of conjugation that we've been discussing. So this passing on of these genes can cause resistance to antibiotics, resistance to heavy metals. It can cause them to synthesize virulent factors like toxins and enzymes or adhesion molecules that make that easier for them to make us sick. So if we were to, I like this meme here, comparing bacteria in 1928 to now, we see this cute little puppy dog, looks comparatively very, sweet and weak compared to this one. And now if we look at bacteria now, they're much, much stronger. So back here it's showing you, oh no, penicillin, because that was it was the first antibiotic that was introduced to the population. And it was considered a miracle drug at the time because long ago, when it was first introduced, there was a lot of people that died from microbial infections. In fact, I think it's like 1 in 5 children died from various infections. So with the advent of penicillin, that number greatly went down, and everyone was super excited about it. But over time, we found that now penicillin is kind of like a joke to many bacteria. So we're seeing this souped up bacteria that says, oh, is that meropenem again? Pathetic. And obviously meropenem is a more stronger antibiotic than penicillin. So basically, again, they're becoming resistant to these antibiotics and being stronger and harder to kill. So that was conjugation. How about transformation? Transformation is when we capture DNA from the environment. So when an organism dies, it's a chromosomes and its genetic material is going to degrade over time. But in the case of bacteria, sometimes they can absorb these chromosomal fragments when a cell releases them from dying. So the definition for transformation is chromosome fragments are released by a lysed cell, and for reference, lyse means ruptured. I didn't do that good job with that. Ruptured or broken down, dead. So when a cell breaks down or is ruptured or dies, its contents are going to be left in the environment. And part of those contents, of course, are genes. So certain cells that we describe as competent cells are capable of absorbing that DNA from the environment. So competent cells are capable of absorbing that genetic material from the environment. Not all cells are able to do that. Only cells that are considered competent. This process is considered indirect because the transfer did not require any special appendages. So previously when we talked about conjugation, that was a direct method of transfer. So this here is indirect. This is useful for a lot of different types of DNA technology, like biotechnology. So what we can do as scientists in a lab is to induce competent cells to absorb foreign genes that are completely unrelated to the organism that we're using, and we do so by inserting genes of interest into a plasmid and then getting that competent cell to absorb it. So if you were in my bio class, then you should hopefully remember us talking about this in regards to how we can biosynthetically produce insulin. So how that's done is we harvest or cut away the insulin gene from a human. So we take the human insulin gene and we insert it into a plasmid from a bacteria. So the little darkened thick area here is going to be my human insulin gene, and the rest of this is the bacterial plasmid. And then we can get a competent cell that is capable of absorbing absorbing genetic information from the environment via transformation. And now that would mean that we have a bacterial cell that absorbed a plasmid that contains the human insulin gene. So now that means that this bacteria will undergo transcription and translation to produce human insulin. So we have a bacteria that produces a product that we desire as humans. Package it, sell it, now you have treatment for people that have diabetes. So insulin is not the only one that this is used for. There's lots of different examples that we could list that utilize this transformation process for human interest. Another term of interest is transfection. This is a similar process, but it's carried out in eukaryotic cells. So we can genetically engineer yeast, plants, and mice, etc., any of eukaryotic organism to produce some kind of desired protein or trait in other organisms, in eukaryotes. For example, if you've taken my biology class, you have seen my zebrafish, and y'all know I can't draw. Or not my zebrafish, I'm sorry, my glowfish. So my glowfish are very brightly colored. They're an example of a genetically modified organism that has a gene in it that is contributed from a different species. So it's sort of like transfection, where we've induced it to produce various colors that it normally didn't, because we harvested genes from, in this case, jellyfish that we now see in these glowfish. All right, so we went through conjugation, we went through transformation. Now we're going to do the last one, which is transduction. Just a note, our book does go into different types of transduction. We're only going to talk about generalized transduction, so don't worry about specialized transduction. It's not covered and it will not be on the exam. So in generalized transduction, the key here is there is a bacteriophage. Remember, a bacteriophage is a virus that infects bacteria, and it serves as a carrier from a donor cell to a recipient cell. So if we look at the sequence of events, we see our bacteriophage, and it has injected its viral DNA into this bacterial cell. Now, what do viruses do? They make lots of copies of themselves and eventually get released. So during replication and assembly of all these various newly made phage particles, it may incorporate a segment of the bacterial DNA by mistake. So now, and this is showing you separated piece of host DNA, this is bacterial DNA, this is all viral DNA. So now what we're seeing is we have 3 little viruses that have just viral DNA, but then we have this one here that has a little bit of that bacterial DNA too. So now what? This cell will eventually rupture, releasing those viruses, and then this virus will go on to infect new cells. So here we have again our bacteriophage that has the bacterial DNA that it incorporated from cell A, and if it infects cell B and injects its DNA, well, now that means that the bacteriophage is injecting DNA from cell A into it. Pretty cool. So it's indirectly taking bacterial DNA from cell A and infecting it with cell B. So now cell B is going to be able to make proteins and genes that cell A did. Pretty neat. So in summary, I really like this table because it tells you basically everything you need to know about these 3 different types of horizontal gene transfer. We said there's 3 types: conjugation, transformation, and transduction. This shows you how it's done. Donor cells with the pilus, the fertility plasmids in the donor. Both donor and recipient are alive and form bridges between the cells to transfer DNA, and it was a type of direct transfer. Transformation, that's when DNA from the environment was absorbed by competent cells, indirect. And then here in transduction, we have to have a bacteriophage involved, which infects cell A, incorporates some of the bacterial DNA into its viral genome, and so when it goes on to infect the next bacteria, the injection will cause the infection of not just viral genes, but also bacterial genes from cell A. And that's an example of another indirect method of gene transfer. Alright, so moving on. Next, we have jumping genes. Jumping genes can be transferred from a chromosome to a plasmid or vice versa. So you can think of these transposons as jumping around inside of the cell. Some replicate before they jump, and some just simply move. So what you're seeing here is the original cell. And here we see a transposon, which is a collection of genes or a gene that can move around. And then you can see some various possibilities. Here we see that the transposon simply moved to a different place inside of the chromosome. In this scenario, we can see it made lots of copies of itself and put it throughout the bacterial genome. And in this case, we see that it was taken out and used to form a plasmid. And then that plasmid could be used to transfer it to another bacterial cell. So how can that affect the bacteria? It can alter colony morphology, it can alter pigmentation of the colony. It really depends on what genes make up this transposon. It can change antigenic characteristics, replacement of damaged DNA, intermicrobial transfer of drug resistance, again, like we keep talking about. There's lots of different things that can help assist the bacteria in increasing variation among the population. So next, we're going to go into 8.5. This describes changes in the genetic code. We're going to be very, very, very brief here. So there's a lot more information in the textbook than what I'm going to cover. But again, remember, I follow state standards, and if it's not in the PowerPoint, it's not on the test. So what is mutation? It is any change in our DNA of the genome that is altered. So any change in DNA. These changes can involve a loss of a base pair, the addition of a base pair, the rearrangement of the order of base pairs, and so on. And just a reminder, remember the base pairs are referring to that order of A's, T's, G's, and C's. If I had a gene and in reality a gene's going to be much longer than this, and I'm not going to draw the second strand because we're going to assume it's there because of the law of complementary base pairing. We know this would be TA, C, GGG, TTTA. But anyway, if this is the strand of DNA that is associated with the gene for, I don't know, we'll go with hemoglobin. So if I were to add an extra base, or delete an extra base, or if I were to insert a new base, all of that is changing the sequence of my A's, T's, G's, and C's. And that's what a mutation is. It will wind up potentially causing a different protein to be made because DNA is transcribed into RNA and RNA is translated into protein. So again, if our blueprints, if DNA, the blueprint is altered, then it's going to change the shape of our house, which is the protein. Wild type organisms, they exhibit natural, non-mutated characteristics, but our mutated organisms we call mutant strains. They show variance in one of the following, and sometimes more than this, but they can have a change in morphology, which is how they look. They can have nutritional differences. They might need more or less things. Their genetic control mechanisms can change. They can be resistant to chemicals that they previously weren't resistant to. It can alter their temperature preference. It can alter enzymatic function. So this mutation causes variation once again in the population. They can have positive effects or negative effects. So these mutations are permanent and they are heritable because genes are transferred from one parent to the offspring. Most of them are spontaneous and not beneficial, but a small number create variant strains that are more readily able to adapt and survive and reproduce. So as long as the environment is stable, mutants only compromise a small percentage of the population. However, whenever we have a drastically changing environment, some of these mutants will be more equipped to survive in the new environment and thus become more prevalent. Again, great example is the acquired drug resistance. That is a very clear model of this type of natural selection and adaptation in microorganisms. Finally, working with DNA in the laboratory, this is our last section. So in the lab, scientists use their knowledge of genes and how things work to their benefit, and that's what biotechnology is. So in this section, we're going to be very brief. My goal is for you to know essentially the function of some of these technologies and not all the ins and outs necessarily of how they work. So restriction enzymes are the first one, and these are also called restriction endonucleases, but they're pretty cool. They're naturally found in bacteria and we can harvest them and use them to our benefit. What they do is they recognize a target DNA sequence and cut it. So for example, this one here, it doesn't give a name, but let's say hypothetically its name is HinD3, because that is one of them. And let's say it recognizes the sequence CTAG and it cuts right before the C. It'll do so on the other side, and now we have cut DNA into fragments. Now, why is that beneficial to us? Because now that we've cut it into a fragment, we can now insert a different fragment into it. So, for example, if we had just in this previous slide here, let's say that we just use this to open up a plasmid. So if I have a circular plasmid and I cut it one time, now I have a linear, I could draw it as a line, but it's this, I have this gap here where I can insert something. And that's exactly how the human insulin gene gets inserted into bacterial chromosomes, like we talked about previously. So we can take this method and harvest plasmids from bacteria and also harvest what we'll call genes of interest, what we want to make, like human insulin gene, and stick it in to the bacterial plasmid. So if this was my human insulin gene and this is my bacterial plasmid, I can link them together now like that. So then I would put it back into the bacteria, and now I have a bacteria that makes human insulin. And so we can have lots of different other examples besides insulin, as listed here: growth factors, hormones, and there's plenty of more. Oops, sorry, too far. So another use for these restriction endonucleases is they yield restriction fragments. So think about this in eukaryotes for a second. And this is a chromosome right here. So if this is let's say chromosome number 12, where we have 46 total, 23 pairs, and I cut my DNA here, here, and here. How many fragments would I have? 1, 2, 3, 4. Now let's say I cut your DNA here instead of, we'll say the first one was mine. I'm sorry, it's not drawn to scale. This is way too big compared to the first one. But let's say this is your number 12 chromosome and this one was mine, and I used the same restriction enzyme. Well, this might be the pattern we see. It cut 3 times and made 1, 2, 3, 4 fragments. But the size of the fragments are going to be different because your DNA is different from mine. But we have methodologies where we can compare those fragments so we can see relationships between organisms. And that's called gel electrophoresis. So you should know what gel electrophoresis does. It size separates fragments. So larger fragments migrate slow. Small fragments are very quick and they migrate quickly. And what a gel electrophoresis process will do, it'll run this DNA that is placed in wells at the top of the gel or the beginning of the gel, and it will allow for an electrical current to pass through it. And DNA is negatively charged, so the DNA is going to begin migrating to the positive end, and it'll wind up creating these banding patterns in each lane based on size of the fragments. So here we see if we have a DNA sample and it cut here, here, and here, we're going to result in having 1, 2, 3, 4 fragments, and they're of different sizes. This one's going to travel really fast and wind up towards the bottom of the gel, whereas this one's relatively larger and will wind up towards the beginning of the gel. Now, this is just showing you 4 fragments, and that's not typical. So if we look at a real, this is real, a real gel electrophoresis sample, you'll see that this lane, I don't know what's going on with my mouse, you guys. We'll see, there we go. We'll see that this lane has, I don't even know if I'll be able to count them, but it has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 fragments. And each of these fragments vary in length from the largest being up here and the smallest being down there. So if I were to run your DNA in 2 of these lanes, 1 and 2, the pattern we see in lane 1 would be the exact same as lane 2. And the more banding patterns we have similarities between 2 different people, the more related they are. So if you were to compare the banding patterns of you and your sibling, and then you had me in the 3rd lane, my lane would look much more different than y'all's compared to y'all's to one another. Alright, next, polymerase chain reaction. Think of this like a molecular Xerox machine. And a Xerox machine makes copies. And that's exactly what polymerase chain reaction does. It uses a thermocycler, which is a contraption that cycles temperature to expose DNA that we want to copy, and it allows for many, many, many replications of DNA to be made. So essentially we can start off with just a small sample of DNA, and at the end of this process we will have a large amount, millions, billions even, of copies in the matter of hours. And lastly, believe, nope, almost last, synthetic biology. This is creating new biological molecules and organisms from scratch. Pretty interesting. So in 2010, Craig Venter created a self-replicating bacterium cell from the 4 nucleotides of DNA. And this was a breakthrough of major proportions, as it was the first time a living replicating cell had ever been synthesized from chemicals. That means he created a living thing from ingredients. That's wild. That's so cool. But this is probably going to revolutionize medical science, because now we have the ability to create precise chemicals to replace those that are missing in disease. We can assemble customized immune components. We can construct biological molecules that can target cancerous cells or pathogenic microbes. These are all potential possibilities that we could see in the near future. Finally, we can also talk about gene techniques and how they treat diseases. 2 of which that are, well, one is used, and there's several different examples of clinical trials. For example, I know that they do it in sickle cell anemia. But gene therapy, this is where we will utilize a virus and alter a virus to where it's no longer able to make a person ill, but it will incorporate a healthy gene into its viral genome that stems from human. So if this was a hemoglobin gene, we cut it out of the human genome via restriction endonucleases. We insert it into a viral genome. So this is the human gene. The rest of it's the viral gene. We allow it to be reinserted into a virus. And then we are going to take a bone marrow sample from the patient with some kind of genetic defect. The marrow cells are infected with the retrovirus that is altered, and the transfected cells are now reinfused into the patient, and the patient is observed for expression of normal gene. So that means, since they have these virally infected cells that contain the healthy hemoglobin gene, they're now going to start making healthy hemoglobin. Pretty neat. CRISPR is another option. I don't believe any clinical trials have been using that yet. I could be wrong, but it is pretty neat. But it's a very interesting methodology that allows scientists to cut an organism's DNA where they want to and insert a different gene. CRISPR is a complex that derives from bacteria, and it has a lot of potential in curing disease, but it's still in kind of its developmental phase in clinical research. So that is it. That is Chapter 8. I hope you enjoyed, and good luck on the well, I guess it's not really an upcoming exam yet, but let me know if y'all have questions.